There isn't a single breakthrough that I can think of that wasn't built on a mountain of failures and persistence. This is a really great example of why funding basic science, or as we like to call it, discovery science, is so crucial, even though you can't see a straight line between what we'll learn from that and how that benefit humans in the long run. It exists. And so I think learning anything about life is always going to benefit life at some point down the road. I'm Eric Wagner. I'm a professor in the Department of Biochemistry and Biophysics here at the University of Rochester. I'm also the Associate Director of the Center of RNA Biology and the new co-director of the brand new New York State Center of Excellence in RNA Therapeutics and Research, or CERT. Our body's primarily made of proteins. That's the working part of your muscles and your bones and your eye and your brain and so forth. RNA contains all the information to convey those blueprints from the DNA to the protein. So if there's any disruption in how RNA is regulated or metabolized, that translates to human disease. Specifically in brain cancers and glioblastomas, there was significant evidence that the RNAs had become shorter, and that allowed the RNAs to translate more proteins, be more stable. It gave the tumors plasticity, basically another pedal that they could just evade growth and just spread around in the brain. We have continued our work to try to understand which RNAs become shorter within tumor cells and understand what that means to the tumor. What advantage does that give the tumor if they have shorter RNAs? If glioblastoma shortens those messenger RNAs, it allows them to overproduce this laminin. That allows the glioblastoma to spread throughout the brain. We've designed these CRISPR they're called dead-Cas13 enzymes, which are tools from bacteria that we can introduce into human cells to force them to re-lengthen those lamin messenger RNAs. If we do that, now we've taken that weapon away from the tumors, and those glioblastoma cells don't spread as easily. For someone who studies basic research in the lab, I don't get many times where I'm out around people who actually have the disease that I'm studying. About 2 years ago, I was sitting on the couch in my living room, and my wife was on social media and brought up a protein called BRAT1. And she said to me, "There's a little boy at my school who has a BRAT1 mutation. His mutation gave him a neurodevelopmental disorder where he needs a walker to walk around. He has some intellectual disability, trouble communicating, but he's a wonderful little kid and his parents are amazing." I always knew kids like Jackson existed, but I didn't sit in a room and hear him laugh and watch him play with toys and realize that I am working on something that could actually help other kids down the road like Jackson. So that was a really powerful moment in my life, to be honest with you. BRAT1's main job is actually to help fold integrator proteins, including one of the critical subunits of integrator called integrator 11. We don't fully understand how those mutations in BRAT1 sort of disrupt integrator function, but we're now starting to understand that the RNAs that integrator is making aren't made properly, but at least we now know what BRAT1 is. It's connected to integrator and how all that is starting to piece together. My lab has a lot of expertise in RNA biology. We have a lot of expertise in biochemistry and molecular biology, but we don't know everything. We don't know how to do every experiment that we need to do. So we rely on the infrastructure at the University of Rochester to help us, the core facilities here at the University of Rochester. They're really top in the country, and I knew if I moved my group here, I would basically expand our footprint. I could do things that I wasn't able to do in other places. They've played a huge role in helping us answer those questions when we're unable to on our own. They want to know, what is the scientific question that I'm doing? What are the systems that we're working with? What are the strengths and the weaknesses in the approach? What are the concerns that I have? They go through all of this with us. And that makes a huge difference because I'm not just getting data back, I'm getting their expertise along with it. My lab, one of its strengths is that we embraced a team science approach. This has become very important in the field of integrator biology, for example, because it's gotten so competitive. No single lab can easily accomplish everything it needs to do in a research study to put it all together. You need experts that are in sequencing technology, you need experts in microscopy, you need experts that can create brain organoids in the labs to study this as it relates to human health. My lab doesn't have all of those expertise wrapped up into a single group, so we have to attract other brilliant scientists to work with us. This was one thing that attracted me to the University of Rochester, and our publication record clearly shows that I benefited from this, my team benefited from this. Persistence is the most important feature in science. You take a guess, you make a hypothesis of how you think something's working, and then you design experiments to challenge that hypothesis. You're wrong quite often, and you have to be good at that. I think all of us think about our careers at the back end, meaning, how would I look back on this and think, did I make a difference at all? And when I retire, I think I would like to look back and think, we stayed the course. And maybe there'll be therapies around based upon our work that will really make me think, I'm glad we just stuck to it.